why nature constrains its polymers, when it does it for functional reasons. Then I will give you two examples, and first is about um, cellulose and or biomass, you would say, and I will tell you about how to create new types of plastics from wood, and how simulations can help to remove the constraints, because wood and cellulose is very tough materials, and it really at this moment prevents us to use it more. Then I will introduce multi-scale modeling methodology, which actually has its 30th anniversary. It was published 1995, and by that time, not so much was mentioned about multi-scale modeling, so we were kind of pioneers there. So I will explain it for you, and as a final example, I will tell you about how to do multi-scale modeling about chromatin and so on. So mm, so mm, so nature is using very much hierarchical systems and multi-scales in its materials, and also it uses highly constrained biopolymers from nanometers to meters. And all this will, of course, significantly impact the strength of these materials. It's very smart strategy, because it besides it makes the material as light as possible, but also it saves lots of raw material and energy. And of course, many creations, man-made creations, like the Eiffel Tower, we see here is based on hierarchies. Uh, and these hierarchical systems, they suit very well to be studied using multi-scale modeling. Uh, Sorry, yeah. So besides that, nature also very brilliantly combines its hierarchical structures with composite materials, and this makes the material even more strong. And one example is the bones carrying our bodies. Uh, they combine both soft and flexible protein collagen and hard and brittle mineral hydroxyapatite. Another example is a spider silk, which is, while it is extremely light, is also impressively strong. So it can be stretched 5 times its length without breaking. And if we scale a spider uh, um, silk to the thickness of pencil, it could, in principle, at least theoretically, stop even a flying airplane. Um, Another example, and really great example, is our hair. So it's primarily composed of alpha helical keratins with a high content of cysteine and sulfur bridges, and this makes it very strong. And it forms filaments and um, sorry, I wonder how I could remove. there is one part of my slice is covered by uh, uh, the partisans, and I cannot really see. I don't know how to do it.
We can see the part of your slide, and below we can see just white space.
Yeah, but I wonder how I can remove it myself because it's in the middle of my screen, and I don't see. Um, okay, so
Oh, now we can see. Now we can see.
yeah. Okay, I continue. So this is the hierarchical structure of your hair, and starting from amino acids, and going to protein, and those filaments and fibrils, and so on. Um, okay, now I find a way. So so what is a polymer? So by definition, polymer is a large molecule which is made up of repeating subunit molecules or monomers, and these are linked together by covalent bonds. And the properties come from their long chain structures and topologies and molecular weights and viscoelasticity and many more. So on the right you see uh, some typical topologies of polymers, from linear polymers to networks. And polymers can be roughly divided to synthetic polymers, which are man-made, and plastics and fibers, we like polyethylene, nylon, and so on. They are synthetic polymers, and they are simple, typically simple, and have a random structures, and then also broad molecular weight distribution. Whereas natural polymers, biopolymers, like polysaccharides, proteins, nucleic acids, they all are produced by living organisms. So biopolymers, unlike many synthetic polymers, have regularly well-defined structures, and this is because they are synthesized using biological templates. So synthetic polymers often they have stochastic structures and wide ranges of chain lengths, whereas biopolymers are monodisperse and have very precise sequences. So here you can see some products of both man-made and by nature-made uh, polymers, which we all know very well. Biopolymers then also are usually biodegradable, and they function within biological systems, whereas synthetic polymers are inert and persistent, and perhaps too much of that. So biopolymers, natural polymers, are found in living organisms, and as I mentioned, polysaccharides are one example of them. So like cellulose, starch, chitin, and it's mainly for structural and energy storage molecules. Polypeptides are also known as proteins, and collagen already I mentioned, and then there are enzymes and many different types of polymers, and they're useful as catalysts and signaling, etc. Then we have polynucleotides or nucleic acids, DNA and RNA, and they are used for information storage and transfer. So biopolymers are tailored for specific roles in organisms, and so these can be like a structural support. So for example, cellulose, which I will talk about later, in plant cell walls, and collagen in many connective biological tissues. Also, for biological catalysis and regulation, and we can mention enzymes and regulatory DNA RNA sequences, also stories like glycogen and genetic information of DNA. So cellulose is a fibrous glucose polymerase, and it provides the main mechanical strength to plants. Whereas the DNA is double helical polymer, as we know, of nucleotides, and it encodes genetic instructions compactly in chromosomes. Proteins are polymers of amino acids folded into functional and three-dimensional structures. Of course, there are proteins which don't have these structures. And this silk fiber of spider, you already saw, it combines strength and flexibility. Okay, so constraint biopolymers in nature, they are functionally constrained, as mentioned, and they are done to fulfill some very specific functions. And many of them are structurally or chemically constrained. And this can be through cross-linking or complexing with other molecules or packing in the specific structures. And these constraints, they can be mechanical, they can be enzymatic, or they can be environmental. So now I will go into these two examples I mentioned in the beginning, and I start with plants and cell wall biopolymers as case one, and then later I continue with chromatin DNA protein complexes. And they illustrate very well how constraints are found in action. Okay, so cellulose and lignin in plant cell walls is built from these biopolymers, which are constrained and strongly cross-linked for structural and defensive functions. So this is very important for plants and trees, so that they can grow tall and can also resist pathogens and so on, so that they don't start to rot in when they're not supposed to do that. So cellulose, then it's a polysaccharide. It consists of beta-1,4-linked D-glucose units. Individual cellulose molecules are long and branched chains, and they are strongly hydrogen bonded with each other side by side, which really gives the strength. And multiple chains aggregate into crystalline elementary fibrils, which can be like 3 to 4 nanometers in diameter, and they contain bundles of 18 to 36 chains. So they have a tensile strength of steel if you normalize it with the weight. So it's really incredible how these molecules can provide it. Lignin in biomass, it's a different type of molecule. It's a cross-linked aromatic polymer, and it's highly heterogeneous, and it's made of phenolic alcohol monomers that polymerize into complex three-dimensional network. So lignin fills the matrix around cellulose fibers, and it is rigid and hard, and it works like a glue between cellulose and hemicellulose. Its random and cross-linked structure increases dramatically the mechanical strength. So there is a very high synergy, and it really enables various types of woody plants to support a massive weight. There are trees which grow over 100 meter tall, and this is all because of this. Okay, so now I go to one more specific problem. I can tell you 2 weeks ago, I was a faculty opponent in Stockholm in Sweden, where Patrick Elf did present his PhD thesis, and it's about creating plastics from wood. And this is a very challenging problem. You just don't do it like that. And of course, as you can see from this article I cite in this slide, he was preferring he was performing molecular dynamics simulations, and this is really a very good start uh, to learn more about these systems. These simulation methods have developed so much during the late decades and years. So I was so impressed to see how he was carrying out simulations same way as experiments. So he had very detailed protocols, and he was looking for specific answers. So before I start this, I just give you a very brief history of plastics. So we have to go very long time back in history. The first semi-synthetic plastic was celluloid, and interestingly, it was actually made from cellulose, and that was 1870. And then a few decades later, it was followed by a fully synthetic petrochemical bakelite. I'm sure you all have seen these old black telephones and so on. But then this then also did mark the dawn of modern plastics. And well, the modern plastic is made of fossil oil, and then the first commercial oil well was opened in 1859 in Pennsylvania. Some major fields were operational in Texas in the beginning of 1900, also Middle East later, and Saudi Arabia. So petrochemical plastics like polyethylene and PVC, they were soon found everywhere after the Second World War, and this was because of their low cost, and they were easy to form and so on. So they basically did transform everything from households to industry. So plastic was used. So plastic. Is plastic a problem? I guess it depends on whom you ask, and do we need to some changes? And I'm sure most of you think that we need some changes, because most plastics, as I said, are made of petroleum, and it's a non-renewable resource, and it contributes to climate change and resource depletion. I guess I sound like Greta Thunberg, but I think there is some truth. So over 400 million tons of plastic are produced every year, and only less than 10% is recycled. So mountains of waste ends up in landfills and oceans, and it has been calculated that that 25 years from now, our oceans contain more plastic than fish by weight. So this is the macroscopic macroplastic problem, but there is also microplastic problem because sunlight and waves break these plastics into small tiny particles, and these microplastics then enter ecosystems and food chains, and they are found in water and soil and human bodies. They enter our cells and so on. So on average, it's estimated that every person ingests 200,000 microplastic particles per year, and we still don't know what the health impacts are there. So this really should convince that maybe we need a change. But what are the alternatives then? Well, well, there's the biomass, and that's a sustainable material resource, and it's renewable and highly abundant. So Earth itself produces 180 billion tons lignocellulosic biomass per year. And so far, only a very small fraction is utilized. Also, unlike oil, we can regrow biomass. So materials made from biomass can be carbon neutral, or at least much lower in emissions than fossil oil-based plastics, and especially if it is produced with renewable energy. And after use, they can often biodegrade or be composted, and this will certainly reduce the waste. And there are already different types of bioplastics and which contribute to a small-scale circular economy and so on. Okay, but there's the challenge to use biomass, and that's the natural rigidity. So cellulose and lignin are naturally very stiff and non-thermoplastic. Cellulose, it doesn't melt when it is heated, and lignin, well, it softens at high temperature, but then it immediately decomposes if overheated. And this rigidity comes mainly from extensive hydrogen bonding and the crystalline structure. Okay, so thermoplastic material can be melted and reshaped multiple times. So this fossil-based plastic is example of that. But most biopolymers like cellulose or lignin, they lack thermoplasticity, so they cannot be processed like conventional plastic. So to make wood-based polymers processable like plastic, we have to modify them first. And this can involve chemical modifications to the polymer itself, or also by adding plasticizers, which are small molecules which then increase the flexibility. And the goal with these both methods is to reduce internal bonding among the molecules, and this will then immediately increase molecular mobility so that this material can flow when heated. Can you imagine heating a wood and it starts to flow? But okay, so increasing the free volume and disrupting dense hydrogen bond networks is really the key in this work. So let's look at the strategy first. Chemical modifications of cellulose, and this is done by opening the rings in the sugars. So ring opening of glucose units is very effective approach, and it converts cellulose to D-alcohol cellulose. And this disrupts very effectively the rigid structure, and more rings are open, more flexible it becomes. And this then results a lower glass transition temperature. That means the temperature where the polymer softens. Also, new functional groups can be attached to cellulose, so they can in turn enhance thermoplasticity. So aldehydes and carboxyls and hydroxylamine, they show clear changes towards thermoplasticity, and they can weaken interchain hydrogen bonding. Okay, so the other strategy was to add plasticizers, and these are small, often non-volatile molecules added to polymer, and they can increase the flexibility, reduce the glass transition temperature. And so plasticizers, they insert themselves between polymer chains, and this then weakens the intermolecular forces. This gives the chains more freedom to move past each other when heated, and then the material softens at a lower temperature. And of various plasticizers, it turns out that the smaller the plasticizers are, better they work. And this is something found in molecular dynamics simulations of Patrick. So the lower glass transition temperature is very important in developing thermoplastic materials. And it's important for processability of this material, and also for material performance, and then also for storage and usage conditions. Molecular dynamics simulations are very useful in modeling cellulose and lignin in their modified forms, and you learn how these polymer chains move and interact and respond to heat and additives. And molecular dynamics simulations can reveal many details you cannot get from experiments. So, for example, how hydrogen bonding network is formed and disrupted in cellulose, and how this ring opening increases free volume, and how glycerol molecule has plasticized and migrates among the chains, and even how water molecules help lignin molecules to slide past each other. So properties like density, stiffness, and interaction energies can be get from molecular dynamics simulations and so on. So how do we get this glass transition temperature from MD simulations? Actually, we can get it from many of the results we normally calculate from simulations. We just have to make a temperature study, and then we calculate the property at different temperature, and we draw a line. And we see that at some point the line changes its direction, and where this happens, it's the point for glass transition. And typically, when we calculate the specific volume or density in MPT simulations, or thermal expansion coefficients, and also diffusion by if you use mean square displacement is a good way, heat capacity, and many mechanical properties like Young's modulus. So it gives you curves like this, and you can identify glass transition temperature there on the row. Okay, so several polymer properties, they change very much at this glass transition temperature, and also this that they become softer and moldable is one of these. I want to mention this is actually more my what we are also doing, because we have also a project with with biomass and cellulose, and where we use ionic liquids and deep eutectic solvents. And they can directly and actually very selectively dissolve lignin and hemicellulose. So they can break these hydrogen bonds and turn wood pulp into a solution so that it can be spun and cast into new shapes. So here is an illustration how it happens. So these actually they don't change the polymer covalently like chemical modifications do, but they provide new processing routes under mild conditions. Yeah, and molecular modeling is very useful in these studies. So let's go to DNA. And so our DNA is tightly packed and constrained by proteins into nucleosomes. And this is crucial to genome organization and gene regulation. So this nucleosome, it provides a natural strategy how to achieve higher order functions. It solves the packaging problem and it creates a regulatory architecture for the genome. And if you want to do simulations here, here you definitely need something like multi-scale modeling, and and so. Uh, So each human cell contains 2 meters of DNA, and then the cell nucleus is only 10 microns in diameter. So how do you put the DNA there? So it has to be heavily, very highly compacted, and nucleosome is just the first level of this compaction, and and then even if DNA is tightly bound in nucleosomes, they are very dynamic structures, and much of that we still don't know so much. And so then linker histone can bind these nucleosomes to facilitate higher order folding, and this creates more constrained structures, and they're called like solenoids or zigzags and so on. So I show you here two figures. There's one comes from multi-scale modeling, and one is from AFM experiments. And you can see that we are already there with simulations. We get very similar results, both from experiments and modeling. So why is DNA constrained this way? Yeah, of course, one is because of the compaction. Nucleosome wrapping compacts DNA already 7 times in this first level, but then it has to be compacted repeatedly, and finally it actually 10,000 times compaction, and that's exactly what it needs to fit in the nucleus. And without nucleosomes, our DNA could not be packed into a cell. Okay, so the other reason for constraining is gene accessibility. DNA around histones is less accessible to enzymes. Okay, so now I introduce the multi-scale modeling method, which is called inverse Monte Carlo, which we did present 30 years ago, and you can use it to do successive hierarchical coarse graining. And this image shows in these colored boxes 3 levels of the all 3 scales of the multi-scale, starting from first principles and going to classical molecular dynamics and then mesoscale. and above you can see how DNA look like at these different scale. So with this method, we can go from atomistic simulations to coarse graining, and we can actually do something like first order coarse graining, what most coarse grain methods do. But this method is so accurate, so we can even coarse grain the coarse grain model. So we do second order coarse graining or super coarse graining, and this will help us in modeling these nucleosome core particles and chromatids. So the principle here is that we start from a very accurate level. We go a level up, and while doing this, we throw away the degrees of freedom we don't need them. And going from quantum mechanics to classical, we throw away the electrons we don't need, but then we have to use empirical force field. From empirical force field to mesoscale, we have to create new models. And this method works so that while you do these accurate simulations, you also are constructing the model to the next level. And you go to the next level, you increase the size 2 orders of magnitude, and again you calculate a new model. So this way, successively and hierarchically, you go up in length scales. And going up from classical atomistic scale to mesoscale, you can throw away even water molecules, so you don't need any solvent. And solvent is the thing which costs a lot in molecular simulations. This is the algorithm. It starts so that you decide the coarse grain beads in your system, and then you calculate the radial distribution function between the points which are centers of the beads. And you calculate potential of mean force, which you iteratively refine, which you can see on this picture on the right lower part. It converges to the original all atom radar distribution function, and when this happens, then this iterative process has converged, and we have a coarse grain effective potential which doesn't need any solvent molecules. So basically, we solve the inverse problem in physics, and this means that we start from results and get the model, but not necessarily in the same level of accuracy. And here you can see an example. There are sodium and chloride ions in the water. So on the left there is the results, which are radial distribution functions, and on the right there is the model. So using the model, we can calculate exactly back the radial distribution functions on the left. But this is not the point to do the full circle. We can also study lipids. The radial distribution function between the beads are there on the left, and when we invert it, we get these effective potentials on the right. And again, when we use these potentials, we get these radial distribution functions, but we also get many other properties, not only radial distribution functions. Okay, so then how this works, you can see from this slide. So here, for example, there are 1,000 lipids started from random configurations, and they very quickly associate to micelle. You can also build much bigger micelles by taking, like you see in the bottom, you put several of bilayers from what you obtain in your simulations, and spontaneously they start to form larger lipids and so on. And observe there is no, this is a water-driven process in normally, but in this method we don't need water because the water is implicitly inside the model. That's why there are these oscillations in its effective potentials, which are the solvation and hydrogen spheres. Okay, so we can also construct consequent DNA models, and there are very many models in the literature. I'm sure Francesca will tell you more about these models today. But this is the particle model we use. It's quite coarse-grained from the very beginning, but we see a small piece on the atomistic level with counter and co-ions there. And we validate these Gaussian potentials, for example, by comparing with experimental Perston lengths of DNA, meaning the stiffness of DNA. And as you can see, we get quite good agreement here. So then when we create study the nucleus on core particle, we need to first construct it atomistically, which is done on the left. So here are 25,000 atoms. There are in the simulation box. There are then all the needed ions and water, and then after coarse graining, we have 1,350 coarse grain sites, and we have the hydrated ions, but we don't have any water because we don't need it. And so then, of course, we don't need water, but we need counter ions and co-ions. And here are, for example, sodium, potassium, chloride, magnesium, and then there is cohex 3 plus. You'll see on the right, and we studied condensation of these nucleus on core particles then using these models. And so of course we cannot use a fully atomistic model for nucleus and core particles because today it would take too long time to simulate and have converse results. So what we need here, we need different sets of coarse grain potentials, and here we need like three types. One is DNA with ions, and these ions are hydrated ions. They come from simulations with DNA and ions with water also on atomistic scale. Then we have DNA and peptides for DNA protein, and then peptides and ions. And in this coarse grain model, amino acids for protein amino acids are grouped into 5 different groups: hydrophobic, polar, non-polar, positive, negative, and glycine. And for histone, the protein in the middle, the core part, we use elastic network models, which are very simple, but still very good and robust models. So here are results from modeling some isolated coarse grain nucleus and core particles, and there are 20 of these particles at different concentration of ions. So you can see there's potassium, there's magnesium, and there's this cohex, and you can see that going from potassium to cohex, the condensation increase drastically. Of course, they should be connected, and in the next step, the simulations are carried out with 12 connected with linker DNA connected nucleus and core particles, and here we have sodium and magnesium ion, and we can see there's different type of folding in the case of sodium and magnesium, and these conformations are more or less stretch or compact depending on the salt concentration. So to finish this this lecture, um, I summarized the last simulation in this slide, so that you can see how powerful this method is, because on the right there's a super coarse grain um, connected nucleus and core particles, and for that we need 98 beads. But then this is constructed from a coarse grain model which contains 28,000 beads. And the original all-atom simulation, which corresponds to this size of the system, it would have 11 million atoms. So there's drastic reduction of the size, and so now I come to the end of my lecture. I just want to thank you all, and I'm very sorry the problem I had in the beginning. It was my problem, and I managed to solve it.
Okay. Thank you very much. Thank you.
So do you have any questions?
Thank you very much for very interesting overview of this topic. And now we can have some time for questions, maybe several short questions, please. If yes, please, Professor Bogdan Lev.
Okay, I have only one question. You use the inverse method solve the problem about the potential interaction, but inverse method have many other solutions, and this task is not correct. What is way to solve this problem?
What do you mean it is not correct?
What do you mean it is not correct?
The inverse task, this inverse task is not correct in the background. Inverse task has the many other solution about the potential interaction.
Oh,
not only
okay.
one.
So the potential is not unique.
Yeah, it's many.
Yeah, but it has been shown that that between pair potentials and radial distribution functions, there is a unique correspondence. So that means that it can be correctly solve the inverse problem, and there are several papers in literature which have shown that it's correct, but they don't show how it should be done.
So probably we can mention here Henderson theorem.
Exactly, Henderson. Yeah, Henderson papers.
Okay, Aatto and Professor Laaksonen, can I ask also the question, maybe more philosophical? And you show a lot of examples of different systems which can be considered as confined systems. And can we say some definition of this term, confinement? So actually we have
okay, so this course project is about confined systems, and I did use constraint, and I don't know if they are exactly synonyms, these two terms, but in my mind, they are, at least in these cases I did show to you. And I mentioned that there were three different types of constraint. One was structural constraints, and then there was environmental constraint, and
I see. So constraint is more general, and
it's more general than confined.
I see.
So only in structural constraint it becomes confined.
I see.
Yeah.